All right, welcome back. This is section 3 in chapter 13. We're talking about meiosis here today. We're going to be comparing it a little bit to mitosis in how the number of chromosomes or the chromosome sets change from diploid to haploid. Again, with normal mitosis in body cells, you are keeping the same number of chromosomes. You can't have that number change. In human being, every cell has got to have 46 chromosomes. But with gametes, sex cells, sperm and egg, that would be too many chromosomes, because when you take sperm and egg and combine them, you're way over the limit. So we need to go through another process of division, cell division, to get that number cut in half so that when sperm and egg are combined, we get back to the ideal number of 46 for a human being. So we're going to be talking about meiosis 1 and meiosis 2. It actually takes place through 2 divisions, reduction division, and that means we're going to be looking at all the stages: prophase, metaphase, anaphase, and telophase through the first division, which we'll call meiosis 1, and then the same series of phases will occur again through meiosis 2. 2 cell divisions then are going to result in 4 daughter cells rather than 2, like we talked. And here's the big important point: each daughter cell has half as many chromosomes as the parent cell. So, in mitosis, you will recall that one of the most important things that happens is the S phase, where we take our DNA and we duplicate it. Getting the chromosomes duplicated means that once the cell divides, we're going to have the whole complementary set of chromosomes going to each daughter cell. But if we're going to do meiosis, where we're producing sex cells, sperm and egg, then we need to have 2 portions of this process where we take homologous chromosomes, pair them up, separate them, skip interphase, skip the S phase, where we again duplicate our DNA, and proceed right along to meiosis 2, where then we take these sister chromatids, separating them and giving us 4 haploid cells, each with half the number of chromosomes you would expect in a normal body cell. So unreplicated chromosomes are going to be going through 2 divisions instead of 1. So this little series of animations are again intended to walk you through the different steps as we go through meiosis 1 and then meiosis 2. Let's say we have a cell with 2 chromosomes in it. During the S phase, we're going to take those chromosomes and duplicate them so that we have 2 sister chromatids. In meiosis 1, homologous chromosomes are going to separate. Haploid cells with duplicated chromosomes then are going to be further separated as we go through meiosis 2. So now we're down to haploid cells here with unduplicated chromosomes. Again, just like mitosis, before we start meiosis number 1, we need to go through interphase and we need to do the process of DNA replication. Each sister chromatid at the end of meiosis 1 is going to be genetically identical and joined at the centromere. The single centrosome replicates, forming 2 centrosomes, and that brings us to
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming
in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have four haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
All right, so again, that little video clip does a nice job of animating everything and working your way through prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Again, when we start this process of meiosis, we are going to copy our DNA. So if we're talking human being, as we exit the S phase, we have 92 chromosomes. Those chromosomes are released in prophase 1. We dissolve that nuclear membrane. We start attaching the spindle to the chromosomes, moving them towards the metaphase plate, the equator of the cell. We get them all lined up, and in anaphase, we pull them apart. Telophase, cytokinesis. At the end, we're talking about two cells, each one having 46 chromosomes. But that's too many. If we're talking sperm and egg, that's too many. So now we need to skip interphase. We do not want to copy our DNA again because we need to get this number cut in half. So we will begin prophase 2, where we take our 46 chromosomes that we have in two cells, two daughter cells, and we start arranging them by attaching microtubules to the centromere. We start moving them around, jostling around, tugging them back and forth like they were talking about in the animation. We get them lined up on the equatorial plate, the middle of the cell, in metaphase 2. Anaphase 2, we pull those chromosomes apart, and now we have formed, through cytokinesis, two haploid cells. Each one has half the number of chromosomes that we originally began with, and that's 46. This is a crucial process in sexual reproduction because, again, we start off with a cell where we have chromosomes, one donated from the father, one donated from the mother, and how those chromosomes get split up, sorted, and put into 4 cells, going from 1 to 4 cells, how we sort those chromosomes really gives us genetic variation, because you got to think of this as a male producing thousands and thousands of sperm cells every day. Each sperm cell has a different combination of these 46 chromosomes that we start off with originally. Okay, so what sperm cell eventually makes it to an egg and fertilizes it is very random, and that is why, again, you look very similar to your siblings and yet very different from your siblings. All right, so again, this diagram is in your textbook. It's walking you through all these different stages of homologous chromosomes, creating what we call tetrads. A tetrad is a bundle of 4 chromosomes. Remember, we've duplicated the chromosomes during the S phase, so homologous pairs get bound together, and we end up with a situation oftentimes referred to as crossing over. As you can see, these little bundles, these tetrads, are really closely intertwined with one another, and oftentimes get tangled up where a chunk of one chromosome gets transferred to another and vice versa. This again increases the genetic variability within a population, thus allowing the species to gradually evolve over time. So when we go back, and a few final notes here on meiosis, prophase 1 typically occupies more than 90% of the time required for this process. Why so much time? And again, it comes back to the formation of tetrads. We have 92 chromosomes. We have this big glob of chromosomes that have just been released from the nucleus, and we're trying to get these homologous pairs aligned gene by gene so that synapses can occur. This is going to take some time. This is going to take a good chunk of time to get everything lined up gene by gene. In doing so, these tetrads, again, will become entangled, and the potential for crossing over definitely exists, which is going to, again, involve some exchanging of segments of DNA. Each pair of chromosomes forms this tetrad that I was talking about, which means a bundle of 4 chromatids, and it kind of looks like an X-shaped region if you look at it underneath the microscope. Metaphase 1, once we get everything organized, the tetrads line up at the plate, 1 chromosome facing each pole. The microtubules from one pole are attached to the kinetochore like we discussed in mitosis. The microtubules then are attached on the opposite end at the centrosome, and anaphase 1 will begin where we start pulling those chromosomes apart from one another. Sister chromatids remain attached at the centromere and move as one unit towards opposite poles. Telophase 1 allows for each half of the cell to be split up through cytokinesis. This is going to occur simultaneously, forming 2 daughter cells. Now it says haploid here, and that is not true. This is definitely a mistake within the book. We have 2 diploid daughter cells at this point. That's not accurate. We are not haploid yet. We are still diploid because of the fact that we have 2 sets of chromosomes in each cell, one from the father, one from the mother. So once we get cleavage to take place and the cell to split, we are going to skip interphase and jump right into the process of meiosis 2, which, like I said, takes us through the same phases that we were discussing earlier: prophase, metaphase, anaphase, telophase, very similar to mitosis. And now, once we get to the end, now is when we will form our haploid daughter cells. A comparison of mitosis and meiosis. Mitosis conserves the number of chromosomes. So in other words, we want to keep this number at 46. We do not want to change that. Meiosis reduces the chromosome number from 2 sets down to 1 set for each cell. So we're looking at 23. All right, so if you flip your book open to section 3, you're going to find figure 13.9. This does a really nice job of summarizing all the details of mitosis and comparing them to the details of meiosis that we have just covered in this program today. I hope this video was very helpful, and we'll proceed on to chapter 14. Thanks.